Hi guys, welcome to another lecture that is part 2 of the cell biology course. I hope you all have watched the part 1 of this course that covers the basic of cell biology. If you haven't watched that video, please first go and watch that. It would be very helpful for you. So guys, coming back to our lecture 2, in this lecture I will be discussing how the discovery of plasma membrane was done and what are the different models which were given to explain the structure of plasma membrane. So coming to the discovery of plasma membrane, plasma membrane is also known as cell membrane, cytoplasmic membrane or the plasmolema. Plasma membrane is dynamic, means it can move. It is fluid and it is also selectively permeable membrane structure. Selectively permeable. So what do we mean by selectively permeable? It means that it can allow the entry and exit of some selective substances only outside or inside the cell. It also forms an external boundary of the cell. It is about 75 m strong in thickness. So plasma membrane is actually about 75 m strong in thickness only. Next, to explain the structure of plasma membrane, various models were given, which we will be discussing in the lecture one by one. But out of these models, Singer's and Nicholson's fluid mosaic model was the most widely accepted model. So the very first model of plasma membrane was the Overton model. This model was given by Overton and hence named the Overton model. So what Overton has observed, that substances which are soluble in lipid can only selectively pass through this membrane. Based on which he said that plasma membrane is made up of lipid, which is a thin layer of lipid. So this model was given by Overton. Overton observed that substances soluble in lipid could selectively pass through the membrane. On the basis, he stated that plasma membrane is composed of a thin layer of lipid. Now, many of you would have been known the structure of lipid molecule, but let me explain it for those who don't know it. So this figure shows the structure of lipid molecule. The lipid molecule have a hydrophilic head. Hydro means water and philic means loving. So it means that lipid molecule have a water loving head. Next part of lipid molecule is hydrophobic tail. So what is meant by hydrophobic tail? Hydro means water and phobic means hating. So this is hydrophobic tail, meaning water hating or water repelling component of lipid molecule. So this was about the structure of lipid molecule. Next come the Overton model of plasma membrane. Here in this diagram, as you can see, the lipid molecules are arranged in a single layer, so to form a plasma membrane. So this model was given by Overton model, as I explained about the Overton model. So our next thing is various model of plasma membrane. Second type of model is lipid bilayer model. Lipid bilayer model was given in the year 1926 by Gottel and Grendel. So इस मॉडल को एक्सप्लेन करने के लिए गॉटल एंड ग्रेंडल ने इरेथ्रोसाइट मेम्ब्रेन से लिपिड को एक्सट्रैक्ट किया था और उनको स्टडी में मिला कि जितना लिपिड एक सिंगल लेयर में प्रेजेंट होना चाहिए था प्लाज्मा मेम्ब्रेन में, उसका ट्वाइस मिला. मतलब लिपिड का अमाउंट जो था वो 2 टाइम्स मिला एज कम्पेयर टू सिंगल लिपिड लेयर. So they thought that it may be a lipid bilayer structure. So they gave the conclusion that plasma membrane lipid bilayer की बनी हुई होती है. This model was proposed to explain the structure of plasma membrane given by Gottel and Grendel in 1962. They have extracted lipid from erythrocyte membrane but found twice the amount as was expected in single lipid layer. So they concluded the lipid bilayer structure of the plasma membrane but could not explain it properly and laid the foundation of future plasma membrane models. Okay. Next come our third model, that is sandwich model. This model was proposed in the year 1935 by Daniel and Davison. This model has two layers of phospholipid molecules. The phospholipid molecule in this model are arranged in a way that the head molecule faces the outer surface of the leaflet while the hydrophobic tail faces the inner surface of the leaflet. This model also suggested that polar head also has a single layer of globular protein attached to it. As you can see here in the figure, this is the lipid bilayer and these are the globular proteins. So globular proteins are attached to the lipid bilayer and forms the sandwich. That is, lipid bilayer is sandwiched between the two protein layers. Phospholipid are arranged in such a way that the hydrophilic head of the phospholipid molecule are facing the outer surface of the leaflet and hydrophobic tails facing inner side of the leaflet. This also suggested that the polar head are associated with a single layer of globular proteins. Okay. So now the fourth model of the plasma membrane, that is unit membrane model. So unit membrane model, also known as protein lipid bilayer protein model. It was given in 1935 by Robertson. रॉबर्टसन ने सरफेस टेंशन स्टडी की थी प्लाज्मा मेम्ब्रेन की एंड उनको ये सरफेस टेंशन स्टडीज में क्या दिखा कि लिपिड के जितना हमारा सरफेस टेंशन होना चाहिए, आइडियल सरफेस टेंशन जितना लिपिड का होना चाहिए, उससे उन्हें कम सरफेस टेंशन मिला प्लाज्मा मेम्ब्रेन के लिए जब उन्होंने स्टडी करी. तो इससे उन्होंने कंक्लूड किया कि अगर हम इसमें कुछ प्रोटीन्स अटैच कर देंगे लिपिड्स के साथ तो उसकी सरफेस टेंशन जितनी हमें इस एक्सपेरिमेंट में मिल रही है उतना हम प्रोटीन को अटैच करके सेटिस्फाई हो जाते हैं. तो इन्होंने कंक्लूजन दिया कि शायद लिपिड बाइलेयर के साथ वहाँ पे प्रोटीन की लेयर भी अटैच्ड होती है. So they have done surface tension studies of plasma membrane and suggested that it was lower as compared to that it should be for lipid only. So they proposed the presence of lipid membrane with protein on both the surface of the plasma membrane. Next come our fifth model, fluid mosaic model of plasma membrane. Please correct it, this is our fifth model, not fourth. So this model was given by Singer and Nicholson. So they have said that plasma membrane do not have uniform deposition of lipids and proteins, but they are formed by a combination of two. मतलब प्लाज्मा मेम्ब्रेन का कोई लिपिड की या फिर प्रोटीन्स की उसमें कितना मिलेगा हमें, उसकी कोई फिट कंपोजिशन नहीं है. इट कैन वेरी फ्रॉम सेल टू सेल. एक सेल में कुछ प्रोटीन्स कम हो सकते हैं, दूसरे सेल में प्रोटीन ज्यादा हो सकते हैं, या फिर एक सेल में लिपिड का अमाउंट कम हो सकता है और दूसरे सेल की प्लाज्मा मेम्ब्रेन पे लिपिड का अमाउंट ज्यादा हो सकता है. दे ऑल्सो सेट दैट मेम्ब्रेन इज नॉट सॉलिड एंड क्वासी फ्लूइड इन नेचर. उन्होंने क्या बोला, मेम्ब्रेन जो है सॉलिड नहीं है और ये क्या है, क्वासी फ्लूइड है. क्वासी मतलब सेमी फ्लूइड है नेचर में. The lipid molecule are present in bilayers. और lipid molecules कैसे arranged है, bilayer में, मतलब 2 layers हैं. जैसे कि हमने प्रीवियस 2 मॉडल्स में भी पढ़ा लिपिड बाइलेयर बनी हुई होती है, वैसे ही इन्होंने भी कहा कि प्लाज्मा मेम्ब्रेन में लिपिड बाइलेयर बनी हुई होती है. नेक्स्ट इज द प्रोटीन मॉलिक्यूल आर प्रेजेंट ऑन बोथ इनसाइड एंड आउटसाइड ऑफ द लिपिड बाइलेयर. अब इन्होंने बोला कि लिपिड बाइलेयर के दोनों बाहर की वाली साइड भी और अंदर वाली साइड भी, दोनों साइड जो है प्रोटीन्स अटैच्ड होते हैं. So as the membrane is fluid, lipid molecule move within the membrane laterally. So जैसे कि हमने देखा कि फ्लूइड है तो लेटरली मतलब साइडवेज, तो लिपिड मॉलिक्यूल क्या है, जैसे कि प्लाज्मा मेम्ब्रेन फ्लूइड है तो लिपिड मॉलिक्यूल क्या करते हैं, साइडवेज मूवमेंट कर सकते हैं, मतलब उसी लेयर में, सेकेंड लेयर में नहीं जाते, उसी लेयर में मूव कर सकते हैं लेटरली साइडवेज. The component being mobile are capable of coming together and form various type of temporary interaction. क्योंकि component mobile हैं वो आपस में पास भी आ सकते हैं और दूर भी जा सकते हैं, लेकिन जब वो पास आते हैं तो वो कुछ interaction हो सकता है उनके बीच, और जो कि temporary होता है. जब वो दूर जाएंगे तो फिर से वो interaction break हो जाएगा. So with this we have completed the various model of the plasma membrane. Next हम बात करने वाले हैं composition of plasma membrane के बारे में. So now let's talk about the composition of plasma membrane. If we talk about its composition, it contains lipids and protein which may vary in composition in different cell types, as I have explained in the semi-fluid or quasi-fluid model. So there are two types of carbohydrates which are present in the plasma membrane. One is glycoprotein and the other one is glycolipid. So glycoprotein होते क्या हैं? ये ग्लाइको मीन्स कार्बोहाइड्रेट, मतलब शुगर मॉलिक्यूल्स एंड प्रोटीन्स में से अटैच्ड होते हैं. सो दे आर नेम्ड एज ग्लाइकोप्रोटीन्स. These are carbohydrates that are attached to the protein part of the plasma membrane. Next are the glycolipids. So glyco, as we know, carbohydrate or sugar molecules, and lipids because these sugar or carbohydrate molecules are attached to the lipid. So these carbohydrates are attached to the lipid part of the plasma membrane. Primary component of plasma membrane is lipid bilayer, which we have already discussed in the model of plasma membrane. Then hydrophobic interaction also play the major role in lipid bilayer organization. Next is a phospholipid bilayer membrane. As you can see here, what we have discussed in the composition of plasma membrane, we can see here. These are the lipid bilayer, first layer and the second layer. So it is known as lipid bilayer. Next are the glycolipids. These are these are carbohydrates attached to the lipid part. So they are glycolipid. Then come the globular proteins. These are embedded in the lipid but not attached. These are embedded. Then is the glycoprotein. So these are attached to proteins, and these are proteins, and these glyco molecules are attached to this protein. So they are named as glycoproteins, or we can say carbohydrate molecules are attached to proteins. Then cholesterol is also present. Then are integral protein. They are embedded fully in the plasma membrane. These are alpha helix protein integrals. So they cross the lipid bilayer. And this is the N-terminal and this is the C-terminal. Next is our peripheral protein. These are attached at the periphery. मतलब ये हमारा बाहर की side attached होगा. So this was about the composition of plasma membrane. We will discuss these phospholipid, glycolipid and glycoprotein in details in the upcoming lectures. This table shows the chemical composition of some purified membrane that are being studied or have been studied. So if we talk about the membrane of myelin sheath, that is nerve cell. So it contains 18% protein, 79% lipid and 3% carbohydrate. Next, if we talk about the plasma membrane of mouse liver, it contains 44% protein, 52% lipid and 4% carbohydrate. In plasma membrane of amoeba, 54% protein is present and 42% lipid and 4% carbohydrate. Next, in human erythrocytes, These contain 52% protein, 40% lipid, and 8% carbohydrate. And the third one is mitochondrial inner membrane. So the mitochondrial inner membrane, I will discuss about the organelle mitochondria in the organelles lecture that will be held soon. So in these mitochondria, there are two membranes. One is outer and one is inner. So here they have studied the inner membrane composition. It contains 76% protein, 24% lipid, and carbohydrate none or 0% they have found. So this was all about the today's lecture. Please watch this lecture carefully. If you haven't understood anything, you can ask me in the comment section, and please give your feedback and I will try to improve myself. So if you like this lecture, please don't forget to click on the like button, share it with your friends so that they can also gain the maximum benefit, and please do like and share and subscribe. Thank you.